Kelsey Hicks.
And I'm Shannon McMinn.
And I'm Erin Zabrowski manning the camera.
Welcome to Baked Goods Immune System.
Alright, so today we're going to be talking a little bit about your immune system. You have two separate parts of your immune system, the innate immune system and the adaptive immune system. And we're mostly going to be focusing on the adaptive immune system, but a little bit about the innate first. It mostly includes things to prevent harmful pathogens from getting into your body, like skin, and then pH regulation and temperature regulation to try and kill things off before they get too far along. And we're mostly going to tell you all about the cells in the adaptive immune response, so we'll give it to Kelsey for that.
The adaptive immune response is directed and preceded by the innate immune response. Today we're going to be explaining the adaptive immune response, which is kind of complicated, with baked goods, cupcakes to represent body cells, and candy to represent other various proteins that are involved in these processes. Hope that we can have a good time with you.
For this section of our video, we will be showing you how the macrophage interacts with the helper T cell to get rid of pathogens. So to start, we have our pathogen and our macrophage, which is a cupcake. Here we have our MHC2 complex, which are little M&Ms, and they're going to interact with the helper T cell. And then in the end, you will see cytokines come into play. Here we have KISS pathogen who has invaded our body, and we are going to show you how to clear KISS pathogen from the body using our macrophage cupcake. The macrophage cupcake is going along through the bloodstream when it encounters the KISS. And we don't know if the KISS is part of the body or not, so the macrophage interacts with the KISS and phagocytoses the KISS. Just like that. And then we take the phagocytosed KISS to the helper T cell to ascertain whether or not this is in fact part of the body. Through the MHC2 complex, the macrophage will give part of the peptide to the helper T cell to tell whether or not it's part of the body. And in fact, this is not part of the body. So the helper T cell releases cytokines, which tell the macrophage to kill, and then it eats the KISS.
Now we're going to be talking about the involvement of B cells in immunity. For the purposes of this part, the green and blue are both antibodies, but antibodies with different active regions and antibodies that work against different antigens. So you also have B cells that have antibodies embedded in the membrane of the B cell, and you also have, same as last time, MHC2 complexes. And here we have differentiated B cells of memory cells and effector B cells. Again, we have a helper T cell, and we will be using cytokines, and in this case, our starbursts are our different kinds of antigens. In our baked goods immune system, we've had some invaders in the forms of starbursts. Here, we have two different kinds of starburst antigens. Here, we have a memory B cell entering this area of the body. Let's see if it does anything. Boop. Boop. That memory B cell did not bind to any of the antigens, so its membrane-bound antibody must not be appropriate for them. Let's see if this B cell does anything. Bloop. Bloop. It's bound to one of the antigens! Now what happens? It goes over to the helper T cell and presents its membrane-bound antigen, which the helper T cell then releases cytokines, which causes the activation of the B cell into both memory and effector B cells as it differentiates and replicates. Now, the memory B cell has the same type of antibody in its membrane, and its reproduction causes a long-term memory of this antigen, which will be useful later down the line. For now, though, the effector B cell begins to produce antibodies which can bind to the antigens, which makes them easier eating for macrophages. In this portion of our baked goods immune system, we're going to be using a body cell, which has a nucleus and it has MHC1 complexes. We also have our memory B cell from earlier, which, though we didn't explain earlier, also has MHC1 complexes available on its surface. Here we have a cytotoxic T lymphocyte, and here we have two different kinds of cytotoxic T lymphocytes which have been differentiated into memory and effector cells. Here we have toxins that can be produced by the cytotoxic T effector cell, which will come into play later. In the normal functioning of the body, sometimes you have cells which become cancerous or are infected by a virus. When that happens, proteins within the cell begin to express themselves differently than proteins in a healthy cell. Now, these unhealthy proteins are then brought to the surface and expressed on the MHC1 complex of the cell, which allows a cytotoxic T lymphocyte to recognize them. Likewise, in a healthy cell, no such unhealthy proteins occur, and nothing is being expressed that a cytotoxic T cell would be concerned with on its MHC1, which is present on all body cells. Now, as a cytotoxic T cell is coming through, if it binds to a healthy cell, then nothing would happen. It's healthy, nothing is being expressed, all is good. It comes along, binds to a cancerous cell. It notes that the cancer cell's MHC1 is expressing the unhealthy proteins, so it begins to divide and differentiate. So it differentiates into cytotoxic T lymphocyte effector cells and cytotoxic T lymphocyte memory cells. Now, the cytotoxic T lymphocyte memory cells serve the same purpose as the memory B cell, but for body cells. So this one can go off and recognize this cancer occurring anywhere else in the body. Now, the cytotoxic T lymphocyte effector cells are going to be used to eliminate this cancer cell from the body. It does that by binding to it and releasing toxins into the cancerous cell, and these toxins cause the cell to want to commit suicide or apoptose. So, the cell dies epically, and then all is better with the world.